So far we have seen a number of different ways of creating biorefineries, different technologies. So we did not discuss range of different technologies, but we focused on those which you are familiar with and which you have seen as a demonstration here. So we'll start with the most topical topic now, which is feed of food proteins. So what we are going to cover is what do we really mean by proteins, and how blue circular economy contributes to alternative protein and how it can be used in different applications. So before we go into the details of blue circular economy, so this just shows what are different types of protein. First one, protein rich food. All the food products which contain higher amount of protein. For example, you have whether sourced from animal, seafoods or pulses, they all are known for good sources of protein. Second terminology what we have discussed as well as you are aware of is protein concentrates. We did discuss about different percentages of protein, mainly either from plant proteins like soya protein, milk protein concentrate, whey protein isolate, seafood proteins or protein derived from fish. Protein isolates, that's again a concentrated form of protein, much purer form of protein. Again it could be sourced from plant, dairy or marine. Or protein hydrosolate, which is produced by breaking down the protein into peptides, polypeptides or protein concentrate. So it gives some example. Cricket flour, that is insect protein, we are aware of it, lot of focus is being shifted towards insects. So one of them is cricket, that is how many of you know this cricket? Figure is correct, yeah, or black soldier fly. So those are the insects, somehow is processed, they are grown on some biomass. When they grow, they are dried and they are milled to obtain proteins. So that's insect protein. Then animal, that's again beef jerky, which is again consumed mainly by the people who look for instant source of protein, for mainly for sports person. Chicken and beef steak. So these are all animal origin protein and a new source of protein. So the differences between isolate, concentrates and hydrosolate is nearly about with the percentages. So there is no clear cut boundary what differentiates between hydrosolates and concentrates. So technically it could be 60 to 80%, more than 90% is protein isolates, and almost 99%, 95% will be protein hydrosolate. So for protein concentration, it depends upon the type of protein, whether it's a rubisco when it comes to duckweed, or alkaline extracted protein from seafood, that is fish, that you have seen how it can be easily extracted, and hydrosolate by using any specific enzyme. Can anybody name one or two enzymes where we can use it for obtaining protein hydrosolate? Which enzyme we can use to get protein hydrosolates? Like, yeah.
Sir, peptides, which break the protein into different amino acids.
Yeah, yeah, it's all can be classified as a proteases, yeah, and then can be a specific protein, endopeptidases, exopeptidases, to breaking which terminal peptide they break.
Yes.
But broadly, papain.
Yes, papain.
Neutral pH. You have alkaline pH 1 alkalase.
Yes.
Or you have microbial sources of enzyme which breaks the protein into specific subunits. But the reason why we use protein hydrosolate, because certain protein is easy.
Enzyme.
Sorry, yeah.
In hydrosolate, we use hydrolytic enzyme. That's protein will be degraded into different amino acids.
True, that depends upon degree of hydrolysis.
Yes.
So mainly in case of improving the protein recovery, once you extract it from alkali, and then if you use a protease enzyme, it can also break certain protein amino acid which is bounding with the starch or with your other biomasses to separate the protein. Yeah, so that's also the reason why we use enzyme to extract a protein. Yeah, so that will become a mixture of protein with different molecular weight. There will be some amino acids, some peptides, and there will be some broken proteins. Yeah, and if you continue that reaction, it will leave it to the peptides or only to amino acids. That's what you mentioned about different peptidases. And if you are looking for a very, very specific enzyme, that specific enzyme will produce a specific type of peptide for a specific application. Yeah. So that's one. So we did cover briefly about protein juggernaut, first generation, second generation, as this is the easiest way to explain when it comes to the phones. Yeah, first generation, making phone calls. Second generation based on plants, that is pork, beef, lamb, poultry, all these meat based one were considered as first generation, and you take this one as an in European context. Yeah, second generation, cereal, pulses and oil seeds, whereas in Asian subcontinent, Indian subcontinent, this could be a first generation. Yeah, because we are mainly reliant on plant proteins. And insect and microbials, cricket, yeast, micro and macroalgae is third generation. Yeah, because these were not mainly used as a source of protein. Mainly used was meat and pulses. Now, fourth generation, that's again in vitro cultured meat, that is lab grown meat, that is all going to be fourth generation.
Sir, nowadays I just listened at the protein based meat, how it will be formed.
Protein
Based meat. Just like animal
Alternative.
meats. Yeah.
Yeah. So that's good question, and it will coming in fourth or fifth slide. You will see how we create alternative meat. So basically, what it is done is normally you take any plant protein, yeah, and try to functionalize it the way it looks like a meat. It is mainly for those who are meat eaters, yeah, and they are scared that they should not convert completely into vegetarian. So they take it as a texturized protein. You would have heard about texturized vegetable proteins. Yeah, texturized, that means any protein, plant protein, can be made into fibrous protein so that it mimics like a meat. So vegan fish product, vegan meat product, and Singapore, there are certain restaurants where you go and order any food product, everything made up of plant protein, and looks like all the texture and everything related to meat. Yeah, so that's the way plant protein can be converted into meat-like texture, not exactly meat, but meat-like texture, and origin is plant. Yeah, I'll show you how we create by using different extrusion technique. And then if you look at World Economic Forum, this particular report is freely available, and it talks about what are the drivers of protein? Why do we look for new sources of protein? You know, you have seen that every half of researchers in well, almost everybody involved in food research, at least 10 to 15% population is involved in alternative protein. Yeah, why do we need alternative protein? What are the sources of alternative proteins? All these are the bigger questions. That's the reason why again we started looking into seafood side of it, or even alternative one, because these could be another alternative for plant based meat, because diet is now shifting from animal to plant. One of the reason is of the climate impact of meat production. Yeah, whether it's a reality or it's a hype, I leave it on to you guys, because that's not the forum to discuss those, because these are all debatable topic. Yeah, land based meat, that means open grazing versus the animal which are grown into beef production, which is happening inside the houses. Yeah, free range chicken versus caged chicken. Yeah, which one is better? So these are all the debates are there, because in Ireland or New Zealand, meat production system is open grazing. The animals are left in a big farm. That's the agriculture. Agriculture does not mean you have a land, you only grow crops. Agriculture also includes animal husbandry, livestock production, and different food purposes. Yeah. Now, if you look at the emission intensity, this is what one of the reason why we are also thinking why people are moving from meat towards alternative plant. Yeah. And you will see some of the statements here coming from different reports. So beef is having highest amount of emissions to produce per kg of meat. So this is cultured meat, Mycoprotein is microbial, chickpea, algae, pork, tofu, pea, jackfruit, beans, insects, wheat, nuts. So, this is basically a standard life cycle assessment method to do and to see how much is having or what ingredient is having more impact on environment. So, this cannot be taken as it always algae will have higher CO2 emissions compared to tofu or pea. It's just based on the type of method and type of product used. So, in order to compare which one is emitting more CO2, you have to develop life cycle assessment model and then to see the real impact on environment. So, this is just the way of classifying the protein, which makes things easy to understand. It is not scientific basis of asking for first generation, second generation or third generation. Yeah. Now, narrative around alternative protein, take it as 3G or 4G, which one is better? Supportive. Food contains alternative protein, lives you healthier life. These all are coming from media and other places, the way it's been promoted at different point of time, whether it's a trade magazine, scientific magazine or non-scientific, non-technical one. Alternative protein are free of risk of food poisoning, yeah, or contaminants. Product based on alternative protein tastes excellent, yeah. There used to be an ad, isko laga dala to life jhinga lala, right? What was the advertisement for? Tata Sky, right? So, it's a similar way, if you will see, there are different advertisement. Another one, better for environment, yeah. Do not harm animal. Obviously, when you are getting a protein, yeah, if you are not killing an animal and if you are eating a plant, you are not harming animals, yeah. That is another narrative. Food security by releasing land currently used to grow animal feed for production of human food. So, certain places animals are grown, for example, beef or pork for animal meat, yeah. So, beef or animal meat, when they are grown and the land is used to grow that animal, yeah, and they are slaughtered for meat purposes. So, this is what it means, that you release a land which is used to rear animals. Do not think what the way animals are grown here, yeah. Animals are tied to a specific farm or a place and they are fed in. Whereas in Europe and other places, animals are moving around in a field, the large acreage of land where they eat the grass, yeah, natural grass, and then only in night time they come back to the room, or out of 365 days, almost 150 to 150 days or 200 days they are outside, yeah. That is open cultivation, grass fed versus grain fed. You would have seen that comparison, that is grass fed, that means left it in open farm to eat grass, or grain fed, that means you make a feed and feed it to animal, yeah. That is the difference, whether grass fed is good or grain fed is good. Lot of research is being going on to say that grass fed is better or grain fed. Now, caution, alternative protein product will always play a minor role in global food system. That is one argument. Whatever we do, alternative protein will not replace traditional proteins, yeah. Alternative protein products are not real food. These are again warnings, and all these things are coming from World Economic Forum report produced by group of scientists led by University of Oxford. Lot of statement you can agree or you can disagree, yeah. Alternative protein products are not as good as real thing. Livestock is more than a food, yeah. So, this whole narrative was against animal food versus non-animal origin food, yeah. So, it also depends upon which side of the coin you are setting, yeah. Either you be unbiased, yeah, talk whether meat is good or plant is good, or if you are completely non-vegetarian, you do not eat anything from the plant, then you will be biased on one side of the argument. Another side, if you are complete vegetarian, you will say meat is not good, yeah. So, we propose both the side. Being a scientist, you have to come with a scientific fact, not narrative fact, yeah. You would have seen lot of debates are there, you know, people they take one side and they just stick to it, yeah. rationality is missing from every debates. Yeah. My line of argument is everybody turns vegetarian. Yeah. What happens to the other side? Yeah. So you can't change the eating habit. People, they have different tastes, different choices. Now, drivers for alternative proteins. Yeah. One, movement of consumer from vegetarian, vegan and flexitarians. Have you heard this word flexitarian? Flexible. Vegan, everything vegetarian. Yeah. No dairy, nothing. No animal origin. Vegetarian is mixed. Vegetarian plus dairy. Yeah. Less input cost, water, energy. To produce a plant protein, we need less energy compared to meat. Because to produce 1 kg of animal meat, animal has to consume at least 50 kg of grass, yeah, or 50 kgs of grain. And to produce 50 kg of grain, you know how much inputs are required. Don't take it exactly 50 kg, but that's the way the things are calculated. Yeah. Muscle building and maintenance, land requirement, weight loss and management. That means if you eat a plant-based food, you lose weight. Yeah. Reduction in greenhouse gas emissions. That means if you are eating plant-based food, there is a reduction in greenhouse gases because you grow more plants and they are the one which sequester CO2. Bold commercial claims about the environmental sustainability. Yeah. I make word bold commercial claims. So, Meet the Future series, Alternative Protein, World Economic Forum. This is the report. It is a white paper document which is available online. The moment you just put this one into Google, you will find all those narrative texts. Yeah. It's very interesting. It's both the sides are written, right. So, it's a niche food or mainstream food. Now, read this advertisement. We make, I am not writing the product here, we make our product entirely from plant without the destructive impact of livestock, yeah, so that you, your children and your grandchildren's children will always be able to enjoy a good old fashioned burger. Yeah. This is a tagline of the one of the commercial product. Yeah. So, I removed the name of the product, it is there. So, how true is this statement? Yeah. I leave it on to you to decide. You all are scientists, you all have studied science. Yeah. It is something like you remove all the lions and tigers so that poor deer can survive. Yeah. So, what will happen? The deer population will increase. So, there is an ecosystem. Yeah. Everything is controlling another species. Yeah. So, there is a role of every system. So, how we deal with it, how we meddle with it, that depends upon humans and what argument we put in place. Yeah. Now, so this is all how the alternative protein business is going one side of the other. Yeah. Because pulses were consumed in Asian subcontinent for ages. Yeah. But now, alternative protein pulses, if you will see a lot of research paper, novel protein from chickpea. Chickpea was always consumed here, right? Alternative protein from mung bean. So, mung bean was consumed not now, like it is being consumed for hundreds of years here, but that becomes a novel source of protein because that was not in the traditional diet in certain part of the world. Yeah. So, that is all how we are driving towards alternative protein. Yeah. So, most interest and investment in alternative protein is currently in Europe and North America, and is from the region that report draws more evidence of social science and political economic analysis. Yeah. From where all this alternative protein things are coming in? From West. It's just because the way the proteins are required for feed purposes, animal meat is not making a business, dairy meat is not making a business, then you need to have to be active in a business first to create a narrative. Sometimes rumors are created to test the water, what people are thinking, other one rumors are created to make things happen. No. So, that is all our different argument with alternative proteins. So, whether it is really sustainable or not, that can only be established based on fact. I will give you a simple example. If you take say for example, any meat, okay, I'll put it simple, chicken meat, yeah, to avoid all the controversies, yeah. If you take a chicken meat, yeah, what happens? When chicken is produced, it is all carbon positive, yeah. Like it takes a feed and then it's leaving the feces, nitrogen emission and all those things, it's carbon positive production, yeah. But when it is processed, it is a product for consumption, right. Take an example of seaweed. Seaweed all along is carbon negative because it sequestered all the carbon, yeah. Then harvesting, processing, still you get a powder, yeah. Then you have to make a product, yeah. So this all makes it carbon positive, right. One place the production is carbon positive, processing and waste is carbon negative. Other place you have production is carbon negative and other one is just direct application, yeah. So unless and until we improve the full farm to fork or we do not take food systems approach, it is difficult to establish what is right or what is wrong, what is sustainable, what is not sustainable. That is why the discussion from farm to fork has moved to food systems approach. That means how the inputs are coming in, how those inputs are developed or produced and what are the processing technologies, what are their implication in environment, and then whatever products you are getting, what is their environmental impact. Am I clear in saying that? Yeah. So I am not imposing any of my viewpoint on this, but for that you read this and come up with your own conclusion, what is sustainable, what is non-sustainable, and talk based on facts. Yeah. It is not based on what you like, what you do not like. And already in a day one disclaimer was given, I am vegetarian, right. Yeah. So just taking the central line, what is correct, what is wrong. Same argument whether jhatka meat is good or halal meat is good. Yeah. One side of argument is if you are cutting a jugular vein, yeah, blood is drained, so there is no blood in the meat. On jhatka meat, that means meat is fresh, yeah. So that's there two different arguments are there, and then again aspects are associated with it, yeah. So is there any study which says halal meat is better or jhatka meat is better? And I do not think so anybody will dare to write anything related to that, yeah. Same thing is what is processed food, what is minimal processed food and what is ultra processed food, yeah. There is a NOVA system to classify different types of processing, and nothing is fresh, everything is processed, because washing is a processing, peeling is a processing, or whether it is less amount of processing or intense processing, that is the difference, yeah. So every food product in my opinion is processed, because processing definition is any change to the raw material is known as processing, yeah. You pluck a mango or you pluck an apple, you wash it before you eat it. So you did simple processing, that is washing. Then you wash it with the chlorine, minimally processed. Then you make a jam out of it, whether it is really heavily processed or minimally processed, or you make apple juice out of it. So that will be less processed, yeah. So there cannot be again a borderline. So there is still a debate whether food is ultra processed, minimally processed or unprocessed. I do not think so anybody will eat unprocessed food, yeah. So wheat grain is unprocessed grain. Who eats raw wheat grain? You either roast it or make a flour, that is also processed, and then you make a bread that is processed, and a bread to burger, yeah. So there can be a lot of arguments. So this could be a best debate to have with all the different type of people. They all are saying their own viewpoint and come to the conclusion, yeah. So why alternative protein? Now if this is the list of the companies involved in this, and it is going to be the third biggest market by 2054, this is what has been projected by business reports and business forums. Companies involved in raw material production, all these companies are involved in production of novel alternative protein. As you can see here, wheat protein, that means gluten protein, yeah. Algae, Aspire, all these, there are only few list of the companies. There are multiple number of companies who are involved in production of raw material as an alternative. As you can see, chickpea is also a company, Nutrati, soya protein, Nordic soya, Cargill, all this pea protein, cultured cell media, and computational breeding and engineered protein. So, these all are the companies involved in raw meat. Processing, texturing, that is extrusion, all these companies are involved in processing aspect of alternative protein. Buller is having big in insect protein nowadays, yeah. Formulation, that is another big area of formulating a protein, having all the nutrients and everything in it, yeah. That means having a balanced amino acid, having all those minerals or vitamins which are delivered in animal meat product system from plant protein. And products, this is again a big one. If you go in KFC, McDonald's and all, you have seen plant based burger, yeah. It's, I don't know how many of you would have tasted it, or plant based sausages and all. All these companies are involved in this particular business also, and lot of new startup are coming in this area as well, yeah. So, you can know like what is a big market, and sometimes research is driven by market or driven by consumer, yeah. It's consumer driven research, but consumer is driven by advertisements, yeah. So, first you create a need and then work on a need. First you create a problem, then propose a solution, yeah. So, that's the way it is. There is no problem, create a problem and then propose a solution. You propose a solution because you have your vested interest in it, yeah. Make sense? So, there can be number of different examples related to that. So, this is all what you can see how different companies are involved. Have you seen Impossible Burger? Yeah, read their statement, you will find that answer, yeah. All these Alpro and all protein innocent, protein rich beverages, juice with protein, yeah. How much amount of protein is required? So, that is everything, even the calorific need has been drifted towards alternative protein. So, this alternative protein is either for food application or for feed application, yeah. So, we are pretty much clear type of different protein, yeah. Hydroslate, isolate, concentrates. What is changed in this slide compared to the previous one? Previous one was having 80, here is 70 to 80, yeah. Now, level of processing, yeah. How we can obtain concentrate depending upon the biomass. Suppose you have seafood side stream, you have done your experiment alkali, yeah, alkali solution, add HCl, you precipitate protein, yeah, you get concentrate, yeah. And if you further purify it, you get isolate, yeah. And then within that there are certain proteins and non-protein items are there. If you take same fish, you add alkylase enzyme, yeah, and whatever you get in a solution will be all protein, peptides and everything. Take the liquid portion up, dry it, you get protein hydroslate, yeah. Or else you get a protein, once you get this protein isolate or concentrate, use your enzyme to develop protein hydroslate, yeah. The only difference between protein hydroslate versus isolate or concentrate can be, hydroslates can be used for making a beverage formulation, because peptides and amino acids will be easily soluble in a water compared to protein isolates or concentrates, yeah. What is the maximum amount of protein you can dissolve in a water? Any idea? Have you ever noticed how much protein you can add in a water and it still remains a solution? If you exceed 4%, you become millionaire, because you can't have more than 4 to 5% protein in a water. That's it becomes completely gel. It makes colloidal suspension. But there are certain techniques how it can be, we can increase the protein. One of them is by using hydrosolate. Yeah, okay. So that's protein rich beverages, 4%, 5%. Protein rich beverage, best one is milk. What is the protein content of milk? 3 or 4%? 5, 6? Mike?
3.5 to 4.5 percentage.
Have you seen any milk which can have 5 or 6% protein?
No, no.
No, only fat can be. Yeah. So that's why any protein soluble in the water, buy any protein isolate or concentrate and see how much protein you can mix in the water. If you get 10%, your process, you are a millionaire. You can make huge amount of money, but it is not possible. It is possible, but it will be like you have to then eat it. You can't drink it. Yeah. So that is one how we can increase the solubility of the protein. Another challenge comes in here. Somebody was asking other day the question about the bitterness. If you make protein hydrosolate, even it is having 10% or 12% protein, but it becomes bit bitter because of certain amino acids. So flavorzyme is one of the options. Other one is to encapsulate that portion of the amino acid. So again, we are making ultra processed product, or we have keep processing it. Yeah, that's one thing. Now coming back to different value chains. Yeah, so protein, fiber, bioactives, and different nutraceuticals, pharmaceuticals things you have seen it. Yeah. Now if you take an example of seaweed here again, harvesting and all with the production, level 1 processing, washing, milling, drying, and different checks. So first level of processing, and then from there itself we can have a product directly for food and feed application, right. Second level of processing, that means converting them into a specific ingredient, whether it's a cosmetics, nutraceuticals, pharmaceuticals, and packaging, processing, and retail, right. So now if you'll see different type of economic value which we can obtain from this value chain. Yeah. Energy, lowest one we already discussed. Use whole of the seaweed as a fertilizer. That will be completely carbon neutral fertilizer. Yeah, because there is no chemicals required to produce that biofertilizer. Food, feed, cosmetics, nutraceutical, and pharmaceutical. So raw seaweed, carbohydrate, fermentation, biofuel, bulk, different types of biochemicals. Chemical conversion, again we can have either fermentation processes or chemical conversion, because even if you take a seaweed, have microbial fermentation or yeast fermentation, you can produce a range of different compounds. Protein for food, cosmetics or animal feed, minerals, food or feed product, vitamin, minerals, you can get different mixtures, residues, energy source, and fertilizer. So next coming slides and tomorrow when you talk about the business model, some of those how we have established large scale system by using multiple different companies. Dultra Marine, this is the organic seaweed producers using IMTA system. They process range of different products directly from seaweed, and this is what produces a lot of biochemical for different application. Now I'll quickly go through different value chains considering blue circle economy, focusing mainly on macroalgae of it, not on fish or chitosan. We have done enough of those examples. Yeah. So this you remember biorefinery. Yeah. I wanted to have it yesterday, but was waiting for you guys to finish off your assignment. You have seen this already anyway. Yeah. Now first one is production. You can fit in any crop into this. Yeah. Example here is again with the 3 different specific seaweed. Then you have a conversion system, that is fractionation, either a dry root, that is you have a biomass, you dry it, do direct extrusion for feed formulation. VC1 is one value chain like this. Yeah. Second one, same dry or wet, both. Either you use a technology to dislodge the biomass to make it a complete paste or to break the structure, and then it can go for food value chain in relation to soup mixes. Yeah, because seaweed based soup will smell like a fish, or blending with other biomass. bakery product, lot of bread and different snacks are produced, or any ready to eat product. And this can also be achieved by extrusion. Seaweed extruded, extrusion changes the properties from insoluble fiber to soluble fiber in that fashion, and residue whatever is left over from these processes can go for biogas or bioenergy production. Same hydrodynamic cavitation from wet or a dry, you have food additives, extraction of protein, alginates and dietary fiber. This you have seen it in on value chains. Yeah. So, leftover biochemical mannitol sugar via fermentation, bioactives, fucoidin, laminarin, antimicrobial agents or different antioxidant. Yeah. So, this all the products what can be obtained from a single biorefinery of seaweed, and it is not a theoretical one. Lot of businesses now are thriving on full biorefinery. Yeah. Now, in order to see what one is the best one and what is economic value, you have to carry out LCA, LCCA or techno-economic assessment, that is feasibility study, that is advancing the TRL level. So, then how you do it? Economic, social and environment. Economic for making profit, social that people should buy it, yeah, should be convinced to buy those product, nutrition wise, health wise, economic wise as well as socially or culturally wise, to change the mindset that yes, this is better product for their health and wellness. Yeah. And then bearable, equitable and viable. Yeah. So, viability of a business having both environment and remember we were talking about 3 different E balances. Yeah. In that way I did not cover to balance the social aspect, which is very difficult to balance it. Yeah. You cannot make a product which is balanced for every society, because there is always needs and wants differs depending upon the consumer. So, first value chain, whether it is a food and feed, is related to extrusion. Yeah. And extrusion, what all we can do. How many of you know about extrusion and how many of you have seen extruder? Okay. So, extruder could be single screw or twin screw. Yeah. And extruder is the one which is also used for making texturized vegetable protein. So, extrusion, whole seaweed goes through the extruder, whether you blend it with other grains or you do not blend it with other grains, you get it straight away as a feed formulation for cattle or fish feed. This is one of the common way of making different type of feed, or same seaweed for insect feed and then insect feed or chicken feed, and this insect itself can be used for feeding chickens. Yeah. So, this is the way you can include circularity, or same insect for fish feed. Or then the same insect is blended with the seaweed to make different combination of feed. That depends upon what is your target animal and what is the rationing, that is how much protein should be there, what all different types of nutrients are there in a particular feed, whether it is a pet food or it is a fish feed or cattle feed. These are very, very clear what is animal requires in terms of nutrition, and then based on that you do a experimental design to develop that feed. One of the best way of developing those type of mixture design is having a mixture design. Yeah. If you is a sort of some of you would have seen, a majority of people they use RSM. Yeah. Response surface optimization. This is one of the technique for response surfaces, where you mix 3 ingredient, whatever output you get, based on that you set a target what you need, and then you get exact raw material required to formulate a product. And the other day somebody was suggesting if we can use 2 different all the shells together to extract chitosan. Yeah. Similar way you can mix number of different biomasses. If you know how much protein is there and what will be the quality of the protein, do some bit of calculation, mix those biomass and then extract the protein. Then you do not have to do the blending. Yeah. That is also possible. Either you get a plant cereal protein and pulse protein and then blend it for amino acid, then you have to mix it properly, or else blend the raw grains directly first and then do the extraction. That is also a possibility, so that whatever protein you get a blended property, a blended protein with all those nutritional properties. Yeah. Extrusion is one. So, I will show you a short video on extrusion, which is done with our one of our PhD, or residue protein alginates dietary fiber. Yeah. This is all food and feed value chain. So, any question related to this food and feed value chain? Now, if you have to change any of the biomass from seafood or shellfish, certain elements will change. Yeah. For fish, you do not need to go for fish protein, you do not have to go for extrusion. Yeah. For food purposes, fish itself is a product, fillets are sold, whereas the protein from fish can be used for extrusion for making animal feed or a pet food. Yeah. So, accordingly different technologies are adopted to create value chain. So, there are different value chains. Now, again for biochemical, nutraceutical and pharmaceuticals. So, once you have leftover biomass, the residue coming from this particular value chain, from food or feed value chain, and you have soluble fraction, you deploy a membrane system, high molecular weight fractions, fucoidin, laminarins and amino acids, low molecular weight sugars, mannitol and other fluorotannins or some sort of biostimulant. When we say biostimulant, these are again the nutrient which activates root growth. So, once you have this, cytotoxicity, human intervention trials and then application. Residue over here, these 2,3-butanediol, succinic acid, these are some of the chemicals which are listed to replace from the biological origin. Normally, these chemicals are obtained from chemicals, how we replace it from biological origin. Yeah. So, this is again related to bioactive, fucoidin, laminarin, residue, human intervention trial. So, how you can create different value chain based on a single technology. Yeah. But that does not mean these value chains can only be created by hydrodynamic cavitation. This was just an example because I showed you those some of those technologies previously. Yeah. So, it could be any of those extraction or conversion systems. Now, just watch this this presentation and this answers your question about extrusion. Yeah. I leave it with the music on. Now, this is a simple small extruder. Yeah. And this shows how different plant protein or seaweed or any sort of protein can be used. Alternatives developed using plant concentrate derived from different plant sources. Yeah. So, when you are extruding, you can add multiple ingredients, vitamins and nutrients, whatever is required or whatever is your desired color or so. Yeah. And then in extrusion, what happens, different ingredients, they are mixed, and this is basically a twin screw extruder. Yeah. And this is the same vegetable protein, how texture has been changed for it. Yeah. We need to create certain types of fibrous protein. Yeah, because meat is having a fiber. Yeah. HMMA is high moisture extrusion. Yeah. Now, this is a commercial scale extrusion. Yeah. This particular extruder is used extensively by Cargill and multiple multinational companies at our place to develop different types of texturized vegetable proteins. Yeah. So, all the configuration and different types of protein are based on different types of dye which are used for making that particular protein. Yeah. So, this is again a texturized one, looks like a completely like a chicken type protein. Yeah. Now, if you are looking for, and he is the one who has just finishing PhD and has done a lot of work on extrusion and converting those texturized vegetable proteins into using extrusion. Yeah. Now, when we talk about extrusion, yeah, what do you think how we can make vegan fish like product? If we have to get a flavor of fish in rice protein, yeah, or how we can create a fibrous protein? Can we create a fibrous protein from any flour? I'll give a simple example. How many of you have seen patisa, the sweet, son papdi? Yeah, seen son papdi? It's having a lot of small fibers. How it's been created? Yeah, constant churning and movement. Yeah, there is no extruder used for that. Yeah, same thing which is a rice with acid. If you take high waxy rice and have a acid in it and keep churning, you need to do a lot of churning, and you will see the fibrous or very spongy or rubbery type structure. Yeah, and then to use seaweed blanched water to get that particular flavor. Yeah, to get a fish type flavor. So, you can have a salmon or mackerel fillet looks like vegan. Yeah, same way if you have certain vegetables or crops, like even a jackfruit structure is very fibrous one. Yeah, so you need certain type of fibers and to have that protein, and when you texturize it, they should be able to hold the moisture. Yeah, because the juiciness is also important when it comes to meat product. Yeah, in order to do that, you have to have certain fibers or carbohydrates embedded into that matrix, then only it can have your good water holding and oil holding capacity. So, there you need to have a protein concentrate rather than going for only protein. Yeah. So, these are the different ways how you can make things texturized. So, extrusion is one of the most commonly used method, or is a possibility to scale up. So, as you have seen in the last how the product looks like, you can create a fibers. Yeah. So, that's did this answer your question how you can create a fibrous protein? Yeah. Extrusion is simple one, other one is high moisture and temperature churning processes, it creates into different fibers, you get fiber like texture, or you have different blends and added flavoring agents. Other one is a stock solution. Even some cases animal stock solution is used to impart a flavor, but meat is coming from protein. Yeah. So, now coming back, quick recap again related to value chain. We talked a lot about different seaweeds. See, Gelidium looks like this. Yeah. Alaria squalenta looks like this. Escophyllum nodosum looks like this. Fucus vesiculosus saccharina latticema. These two are heavily aquacultured. This is wild harvest. This is wild harvest for fucoidin rich, and this is for agar agar, Gelidium. Yeah. Use different technology, you get agar agar alginates. Why I am coming back to these ones? Because these are also the ingredients which are used for improving the water holding and water binding properties. Then when it comes to the extraction and where all these ingredients are located, and if you will see different structure of the crop, this is not scanning electron microscopy, you will not be getting like that, but it is just by studying the structure. As you can see, there is cellulose microfibril, glucans, protein, vulvan, this is from vulvan means vulva, interaction, ionic and strength. So, all these are interrelated. Yeah. Other day we are asking about the question of using ultrasonics or cavitation, how do we get things extracted. So, if we, these are the complex structure, if you want to extract for example protein, protein is somewhere here. Yeah. It is bounded with different sugars and different nutrients. That is the reason when we extract a protein, even though protein is colorless, basically you get normally a white powder, whatever is not color, whichever does not contain a color looks white. Yeah. Because the moment you add it into the water, it becomes transparent, yeah, or translucent. So with this, there are a lot of different sugars are there. If you have to use a solvent, so you have to break that particular network. So remember extraction and valorization principle, this one-liner principle, take what you want or remove what you don't want, yeah, correct? Open a packet, take the chips out, you are taking what you want, yeah. Peel the orange, throw it, so you throw away what you don't want, yeah. That decision is made based on what is in a larger quantity. If protein is smaller quantity and you want to use the residue, accordingly the processes are done. Demineralization followed by deproteinization, deproteinization followed by demineralization, all these actions come into play. So this just shows how these are quietly complicated networks: cellulose, glucans, fucans, glycoprotein linkages, phenols, iodine, alginate network. Now this is to do with alginates. So that's what it is giving a strong structure. So what is the compound which gives a structure to a plant cell? There is a single compound, right?
Cell wall, sir.
Correct. What is the compound which gives the cellular?
Cellulose.
No. Have you about there is a middle lamella? Have you heard of middle lamella? It contains calcium pectate. The moment you add a calcium to any vegetable crop or vegetables, it becomes turgid because the pectic substances are there. Well, the cell wall, it does give a structure, but it gives it turgidity. So middle lamella, that is calcium pectate, that is pectin and a calcium network, that gives the strong structure. The same way alginates over here provides a structure. When alginate is gone, then it becomes easier to break it down, yeah. So if you see this normal seaweed, any cell structure, these are your normal cells, and you have a beta glucan, that is laminarin, yeah, and this is what it looks like, much simplified way. This is fucoidin, yeah, and then you have alginates, alginate layers, you have protein, hemicellular celluloses. So this is the biomass, but you are trying to extract something, yeah. So you know it's not that easy, put any solvent and extract it. So first and foremost, to design an extraction thing of valorization is to target what compound it is, where it is principally located, and what are the solution which it can dissolve with, and then accordingly you can draw down your extraction principles, yeah. Then once you have these ingredient, yeah, then you go with all your properties, whatever application you want to use it, yeah. Now you have seen ultrasonic treatment other day, and you have seen some of the scanning electron microscopy. This is another one, I was trying to find it out from exclusively from seaweed. This is from Escophyllum nodosum, 200 micrometer SEM image. This is the surface it looks like, yeah. Seaweed is normally very smooth. The moment we give some bit of treatment, and you can see some of the pores are formed, yeah, and these pores were very clear the other day when we were looking at the meat structure, yeah, because that was precisely taken scanning electron microscopy. Taken imaging itself is a complicated phenomena. So when we try to dry those biomasses, seaweed and all, and then they just changes their structure. So as you can see from here, all these pores, whereas you don't see any of those pores here, and then when you go closely, and then you can see there are pores formation. So that's the reason it improves extractability, yeah. Now again, agar from gelidium, different techniques we have gone through. Alginate from saccharinal latticema, same technique, the powder you get, and then how you have obtained using different techniques. Optimization, and once all these ingredients are obtained, then subsequent operations are carried out to measure which one is having more impact, which one is having less impact. And all these ingredients are also useful in making alternative feed as a water binding, yeah, or as oil binding, or to provide a specific type of texture, because they have very good water absorption capacities. So again, valorization, this we have covered, yeah, different strategies, you get different extracts, purification, application. Then different types of biomasses, crude alginate, pharmaceutical grade, sugar, peptides, purification, all these things are repeat. That means, once you get your hydrocolloids, or you remove the protein and then how you can get rest of the bioactives, or you remove these bioactives and then you get a protein. Yeah, I am trying to just go for quick recap of all those things what we have done and how we can use it into feed application, and then next one for your biopolymer application. Now, once we have taken all the protein, alginates, bioactives, everything out, left over is your fiber. Yeah, fiber, that means it contains lot of cellulose. Now, this is the way how we can get a cellulose. Yeah, either from the whole biomass of leftover residue. Leftover residue, you do not have to go through the bleaching process or so, then you just go for alkaline treatment of the residue again, whatever is left over, to obtain cellulose. And a similar model can be applied when it comes to rice straw. Yeah, either it's rice straw or any other grain straw, how we can get your cellulose. Yeah, and you all know what is the application of cellulose. What is it? Making biopolymer or alternative plastics, yeah, alternative packaging material, chitosan, cellulose, and whole of the biomass itself. And how we can use whole of the biomass itself for packaging application, any idea? Thermoforms. Sorry, can you give a mic to?
Sir, we can form polychar like that and polyform thermocol.
One is thermoforms, so like what we make, plastic cups and paper cups. You have seen those, paper plates and paper cups. That's the whole biomass exactly can be used, or the specific ingredients can be used, or the leftover biomass can be used to making those type of paper cups. That's
sir, activated carbon we can form.
Activated carbon is another biomass. That means you heat this on it and get that black carbon. And other day I was discussing, most of the toner inks are basically a carbon. Activated carbon is one thing, but normal carbon itself is another one, which is used as a ink for lot of the printers.
And sir, carbon dots we can form with the help of rice straw.
Carbon dots, yes, it can be, yes, all these are now high end applications are there. Yeah, so at the moment itself the whole biomass, it's not valorized. Yeah,
Okay.
there can be some of the niche application, can be very high value application, can be low value application. The whole purpose here is to show what are the enormous possibilities are there with the biomass. Yeah, so whether we target for food or a feed, still a lot of biomass left over there when you purify the protein. For instance, if seaweed contains 10% protein, yeah, if you remove alginates alone, the protein from 10 might go up to 25 to 30%. Yeah, so this itself is sufficient enough for a lot of different types of product. It's having similar protein profile, protein content as what you see in pulses. Yeah, so you created into pulses. If you take these protein out, then you have your fibers and soluble fibers, and these fibers again either to get a cellulose, you use it for activated carbon, or you use it for your making carbon dots or whatever is to be prepared. But how carbon dots can be prepared out of it, I have never done it. So I have I will say zero experience in making that. Yeah, okay.
Sir, actually my senior doing like he make carbon dots from the rice straw and anything like waste material.
That's great. So the similar approach can be adopted here, right? So when you learn from one stream to another, that is why I always call it translational approach. That is translating knowledge from A field to B field. Yeah, that's the way. That was the purpose of bringing this one into this picture, cellulose, fibers and other one, because it was becoming too foody and food and feed chain. Now, once you have all these ingredients, the question comes in whether it contains antimicrobial activity or what type of activities these extracts have. Some of these what you are seeing here is coming in based on your yesterday's, based on the question and answer session, right. So, whatever extracts were obtained, fluorotannins or bioactives from seaweed, Listeria innocua, E. coli, antimicrobial assay. As you can see, this has been, when we do antimicrobial assay, one of the way is to do it in a spectrophotometer. Take a microbial culture, add it into the media and then add your extract and leave it in a incubated condition and then to see how microorganisms is growing. So, this is what you will see, there are too many lines, this is complete raw data there and it is like this. Yeah. Some they do not grow, their lag phase is way beyond their limit. Yeah. This may be just an error. Yeah. It is not that negative growth, not possible. And some they have normal growth. Yeah. So, that means certain concentration of extract they had bactericidal effect. So, if you include into the product or include into the hand cream, this will not kill, but this will stop their growth. Yeah. Majority of the natural extract they are bacteriostatic or bactericidal. Cidal means killing, static means maintaining the activity, that means not allowing it to grow. Yeah. So, that means guard standing at the gate, yeah, or a shooter at the gate. Yeah. So, that is stopping the activity or killing. So, E. coli or Listeria. So, this shows antimicrobial properties, how it can be done. Other one, once you have extract, you know this having antimicrobial properties or some health properties, then you have to do with cytotoxicity, and then cytotoxicity based on different molecular weight compound. Yeah. That means cytotoxicity for all the proteins and alginates, hydrochlorides we obtain, or low molecular weight compound and 5 kilodalton. So, this shows how much is the concentration required to achieve certain cytotoxicity. Yeah. So, this was all some of the examples. How do we take seaweed from farm, get the food ingredient, feed ingredient, extraction technologies we have covered. We have seen how we can use extrusion to develop different types of product. Yeah. And then whatever bioactives are, what are their sequence of events required to establish a full refinery process. Yeah. Quickly just go through the microalgae process. Cholera vulgaris, Nanoscleropsis, Porphyrium purpureum, this is red color. So, direct product itself, you will see this is all in at not lab scale, above lab scale, in a pilot scale you can say that. Fresh biomass, freeze dried, this is the powder what we get. Yeah. This whole itself is a perfect red color powder. The moment you add it even a 1%, whole water sounds completely red. Same way if you have spirulina, you get a bluish color. So, depending upon the microalgae, you can get different types of color. Then extraction, same process, 1:20 dilution, freeze dried biomass with 20 ml, that is 1 gram for distill 20 ml. Yeah. See the color of the solutions. So, you have Cholera vulgaris, Nanocropsis, and again same purpureum, that is as microalgae. And then next, either use a normal shaking method, stirring method, high pressure employed, plasma, ultrasonic probe, bath, yeah, microwave or pulse electric field. Yeah. And you can have much more number of technologies employed in it. Yeah. So, that is all for treatment purposes. Then what else you do? You centrifuge it, you have seen all these centrifugations. Yeah. And then supernatant and residues. Yeah. So, even after the treatment, after giving the treatment, even then you dry it, you will have a better biological activity. There you are using a technology to release those compound into suspension. Yeah. It is not then you have to purify the compound for different application. Main purpose is to improve the bioavailability or to improve bioactivity of the compound. You take a microalgae, make a solution, give a treatment, dry it again, and then use it for application. You will see the differences in bioactivity. Yeah? That means treatment itself is opening up the structure. That is the advantage using microalgae, because they itself are microscopic. Then biotech compound profiling, cell culture studies, and this is the full series of experiments which was done as a part of research thesis. Yeah? And there's been a lot of people were involved in production of microalgae. One group was only looking into improving the bioactivity, other group was extracting the compound, and then this is the sequence. This is exactly what you do in terms of extraction experiments. Yeah?
I am working in plant-based meat and lug, sir. So it is easy to mimic the quantity of protein, whereas chicken has around 28 to 30% of protein. Plant-based meat and lug, I can achieve the same amount or even higher than the existing chicken. The problem is in quality of protein, that is amino acid. We know animal protein has more amino acid, more essential amino acids than plant. So how to overcome this? How can we claim it is an alternative protein, whereas the quality of plant based is a bit lower?
Yeah. Now, first thing is, even though when you're trying to see the amino acid, that's why I said perfect blend is required. Yeah, blending, that means either to having a co-extraction between all the which contains all the essential amino acids, that is pulses versus rice. The formulation has to be right. Yeah, that's one. Other one is to having those amino acid added to that. That's the reason I gave a reference of mixture design. If you see A protein, what you extract, what is their amino acid, and B, what is amino acid and what is your target amino acid. If you do the back calculation, it will tell you how much approximately raw biomass you need to get a co-extracted protein. That's question number one. Yeah, because if you blend the protein afterwards, you may not get a consistency, a cohesiveness between that protein to obtain that particular quality. Apart from that, there is another challenge with the quality is not only the nutrition part of it. Other one is also to have the bioavailability and digestibility of those two proteins. There again, once a protein structure is obtained, and then if you are giving a certain treatments, I don't know which one will be the best one, that has to be investigated, whether pulse electric field or you are giving electrical treatment or ultrasonic treatment or moisture treatment, hydrothermal type treatment, so that you make that texture and improve its digestibility. Yeah, then only you will be able to see whether it's matching exactly that or not. Yeah, so basically majority of the time when it comes to different type of chicken or meat product, the way it's been processed, what people, I don't know, you will be better one to tell me that, is mainly they feel the texture of eating. It's not the taste. I don't think so they're having much of taste, but it's only because of juiciness and amount of water or gravy it can retain. That is the feel what it gets you, right? Because protein is not having any taste anyway. Yeah. So, and that's the way it can be easily formulated. So, amino acid formulation problem is not severe. The problem comes in when we have your getting the digestibility and availability of nutrient plus other vitamins, fat soluble vitamins or cyanocobalamin, how we match that one. That's one challenge. Second challenge, you asked this question, second challenge is when things are related to complete vegan or diet which does not contain any fat. Yeah, there is a challenge, then how you are going to store your fat soluble vitamins. Yeah, because fat is also essential. It's not you eliminate fat completely, you will be more healthier, slimmer, trimmer and all, right. But the problem is how do you retain your fat soluble vitamins. So, all these things has to be well balanced, and in order to create a texture, there is also some techniques you would have seen, I think in a lot of institution in and around Chennai, they have that called electrospinning. Yeah, and think of the guy who sells what you call sugar threads, big sugar candies, big balls. Those are the what do you call that?
Cotton candies.
Cotton candies, yeah. We used to call it something else, yeah. And then if that is created from the fiber, and I did ask somebody to create a fiber from the protein, such suspension, and those type of fibers can be created from the protein itself. Then you don't need to use a huge amount of technological approaches. Yeah. Any other? No. So what we covered basically was quick of recap, yeah, as well as how we can create it for feed and food protein. So next one will continue with the biopolymer, yeah, and there will be some bit of connections from the previous one, yeah.